right, so the midterm is in 2 weeks. Next week we'll finish up 7. I'll give you that take-home test.
Will we get any sort of like study sheet for the
Somebody else asked about. I will look into that. If they have it, it'll be for the whole ODA deal. Okay. All right. So y'all, we're going to be looking at a field of biology known as genetics. It is a fascinating field of study. We already know about the DNA and what it does for making proteins in our body, right? We've looked at protein synthesis and that kind of thing. Yes. Well, also we know that the DNA is responsible for the traits that we have inherited from our parents. Correct? Yes?
Yes.
Right? Okay. All right. Well, the understanding of that, there's still quite a bit that left that is hard to understand. I will tell you this, in my biology courses at UT when I was there, now this does not count the chemistry classes, right, and the physics, those were hard, but my most difficult biology class was a genetics class. It was very, very difficult. So there's lots of data in there, and we do weird things with the DNA, cross them over. Remember, we have multiple genes for which certain traits may be coded, that kind of thing, okay? But the understanding that we have of traits being handed down to the offspring from the parents were really discovered by this man right here, and his name is Gregor Mendel. And so we call this Mendelian genetics based on him. I don't believe he was alive when he got all the credit for this, which is kind of sad because he put in a lot of work. We're going to watch a little biography about him. He was a monk. So a monk is somebody who lives, in a monastery and spends all his time studying the Word of God, maybe copying the Word of God. His job was he was the gardener. And because of that, he spent a lot of time in the garden, and it really benefited us because he happened to notice some things and get very inquisitive about the garden. Yes, David?
Do I get points off for not wearing jeans?
What?
Stop.
Yes,
Oh.
yes, you do. Just for asking
No,
the question.
the
you
joke maybe, yeah, but not the joke.
What?
Cool.
What was his name? I didn't.
Gregor Mendel.
What did you do?
Mendel. Mendel. Okay.
People say.
He's a nice guy. He was not just a monk. He had given up his inheritance so his sister would have a dowry so she could get married. It's very sweet of him. He gave everything up to become this monk, to do this. So he happened to be studying the plant. He had questions about the plants. He was very inquisitive, right? He wondered why the plants did what they did. And it's just serendipity, which means happy accident, that he happened to study English peas, the pea plant. And the reason that's happy is because the pea plant tends to have traits that are handed down just one trait per gene. None of the weird stuff like eye color in humans. Josh, did you have a question?
Oh yeah. So do you think people that wear glasses? Do you think they get that from their parents? Because both my parents wear glasses, and both their
Okay,
each of
so
their
it
parents wear
depends on what the condition is.
Uh-huh.
So if it's the shape of your eyeball, then that is genetic. You got that from your folks. If it's some other, for instance, I have to wear these readers, but it's only because as time went on, my eye got less and less
It's for looking at
bendable.
small screens.
Yeah.
No, I have to wear mine because I'm farsighted.
So the bottom line to that is some diseases that we have are genetic, some are environmental. Okay, so we'll just have to see. I think next week we're going to talk about some of the more notorious genetic disorders. Okay, so we'll try to get a handle on what some of those are. We're going to get an introduction here to Gregor Mendel by watching a little small biography about him. Oh, hold on. I just turned it all off.
Monk, teacher, and biologist, Gregor Mendel is known today as the father of modern genetics.
Gregor Mendel's experiments on pea plants were fundamental in establishing the field of modern genetics.
Born Johann Mendel on July 22, 1822, in what was Austria and is now the Czech Republic, Mendel excelled at school and became an Augustinian monk to further his education. He was given the name Gregor when he entered the monastery.
The monastery sent him to the University in Vienna, and that gave him the education in science, math, chemistry, and botany that he then brought back to use in his teaching.
Mendel accepted a teaching position at the monastery in Brno after completing his university studies. It was during this period he began his famous pea plant experiments.
Before Mendel people thought you would equally inherit the traits of parent A and parent B in equal proportions.
Mendel experimented with pea plants and found that instead there were dominant and recessive traits in all organisms that were passed on statistically based upon the mixing of genes between the two parents.
This theory is now known as the law of segregation. Mendel also formulated the law of independent assortment.
The law of independent assortment addressed the fact that there are a large number of traits. These traits are passed on randomly and are independent of the other traits and of the traits of the two different parents.
The ability to explain hereditary diseases and to engineer plants with desired properties are all consequences of the fundamental theory founded by Mendel.
During his life, Gregor Mendel's research was disregarded by almost everyone. It wasn't until the early 1900s, after his death, that Mendel's work was appreciated and now serves as the basis for genetics and heredity.
In his later years, Mendel became the abbot of his monastery and spent less time with his scientific endeavors. Gregor Mendel died in 1884 at the age of 62.
Gregor Mendel's work laid the foundation for modern genetics, for modern gene therapy, and is the basis for some of the most exciting discoveries in medicine currently and in the near future.
How does he know that it's in the near future?
Optimistic, I guess. Yes.
Why did he die like that
died?
early?
At
Yeah,
age
that's
62?
what I was gonna
Yeah.
ask.
All he ate was
Ask the
peas.
same thing.
It was at a time when we didn't have things like medications.
God needed some
You know.
gene work.
Do you have a question? Just one? Okay.
You know,
All right.
right now
So
you
notice how in the video they alternatively called him Mendel or Mendel. It's pronounced Mendel. So Gregor Mendel is the correct pronunciation of that. Okay, so what did he do? He was out in the garden. Oh, at the end when they said he became the abbot of the monastery, that completely stopped all of his work in the garden because he had so much work to do in the administration of the abbey.
Does that mean
Of the monastery.
Does that mean he's crippled?
That was the worst
He
joke
was.
possible.
I missed it. I didn't even get it.
Greg
He's making
Abbott.
a joke about Greg Abbott.
Ah.
Oh, Greg Abbott.
Yeah, because he said Abbott.
Okay.
And his name
All
is
right.
Greg
I didn't even get that until you brought that up. Okay. All right. So let's see.
How bad it was.
So what he discovered, the first thing he noticed, first of all, I will tell you, he was a very patient person. He was very, think about it, if you have to grow plants, that takes some time, right? It's not like the next day they're ready to go. No, he had to wait for them to grow. And so what he noticed, for instance, in this particular case, he noticed that sometimes pea plants have purple flowers. Sometimes they have white flowers. But he noticed the ones that were purple flowers, what he would do, instead of letting bees do the pollination, he would go in and pollinate the plants himself. So he'd take a paintbrush and go from one flower to the next. All right, and so after some time went by and he got the new peas from the plants that were growing, he noticed that every single generation of the plants that he grew all had purple flowers. And he said, okay, here's what I'm going to say. Because I've been doing this and crossbreeding them with purple to purple, purple to purple, all these times, probably, 10 generations or something. That's a lot of patience right there. He said, what I have then is nothing but purple flowers. The gene, he didn't call them genes, he called them factors. We changed the name to gene later. But he called them factors, and he said, what I have here is something known as true breeding. In other words, they only have one trait on which to pass on to the offspring, okay? So but remember, he had to kind of create this group of flowers that were adhering to this, okay? All right, so he found that the purple flower plants always produce purple flowered offspring. We're going to see this big A, that's going to come to be very important very, very soon. All right, so he said, what I'm going to do then is I'm going to take, on my first experiment, I'm going to take what I know is a purebred purple flowered plant and a purebred white flowered. So while he was doing the purple flowers over in this garden, he was doing the white flowers over in this garden. They grew, everything else looked the same except one had purple flowers, one had white flowers. Do you understand? We know the white flowers are completely only white. That's the only factor they have, the only gene they have. The purple flowers only have the purple flower gene. Get it? He said, so what's going to happen if I cross the purple with the white?
It's gonna make a light purple.
That's what he did. That's what his hypothesis was. And he was wrong.
because the purple takes over.
100% of the plants came out purple. 100%. 100%.
Dominant to
That's when he came up with the idea of dominance. He said, okay. He thought maybe there was a mistake. He said, well, maybe there's a mistake. He did it again.
That's a lot of patience.
A lot of patience, and he came up with it again that they were all purple. And he said, okay. So now he wants to do something else. Guess what he's going to do now? He's going to take the purple of the second generation, and he's going to see what happens when those are cross-pollinated. Okay, we're going to see something interesting there. Yes?
What about like the flowers, like the dandelions and stuff? There are multiple colors. Like,
Okay, we'll get to
the
that.
poppies and the
We'll get to that. Those are things, that's something called, this is why, remember I said happy accident? Peace didn't have that problem. That's a thing called incomplete dominance. These have complete dominance. They don't have that problem. All right, so we're going to say now, later on, gang, what we decided to do was we decided to attribute a trait by using a couple of letters, a double letter to show that attribute. So in this particular case, big A, capital A, stands for the dominant trait. Little a stands for the recessive trait. Now, he doesn't know this yet. He's going to take now this F1, that's going to be that first generation after the parent generation. He's going to cross-pollinate those. All right, so the second experiment, Mendel used his first generation to produce a second. So now we're going to have those that are here, all purple, 100% purple. And he said, what happened to that gene? Where did it go? And this is when he this is brilliance right here. He said, I think it's still there. It's just hidden. It's hidden. How can I show that? That's when he's going to cross-pollinate these. So what did he do? He cross-pollinated those. And I switched over from purple flowers and to tall and short. That was another trait he looked at, was tall and short. So let's go back here really quickly. When he cross-pollinated these, what do you think happened?
Well, it's white again. It's white now. He did it with white.
Okay.
He
I think it was mixed.
He did it with white
Purple.
No, I think like there were some that were white and some that were purple.
That is great. He had 75% purple,
purple.
25% white. And he said, aha, it was hidden there. And the only way we could see the white flower was if those genes were distributed in this capacity, that two recessive genes were shown.
Not if they already had
If there was a dominant gene present, the dominant trait would show. Yes?
So, like, that's what you were talking about, was it like last week when you were like, your parents might not have like red hair, but your grandma might?
Correct.
So
It
just
can
kinda
skip a
like-
generation.
But it's still there. kinda?
Yeah, that's twins is a thing that happens with twins as well. Skip a generation. Yeah. There you go. All right, so I'm sorry, one of the other traits that he studied was tall and short, and we're going to see here where we took a tall plant, a short plant, we got 100% tall. Then he took the F1 generation and he got 75% tall, 25% short. What is the dominant trait in tall and short? Tall.
Yeah, that's
So the
what I was
recessive gene in tall and short is short. Understand? All right, so these are the factors that he studied, all right? He looked and he determined by doing lots and lots of study which was dominant and which was recessive. So tall is dominant, seed shape being round versus wrinkled. Round was dominant. Seed color, yellow was dominant to green. How many yellow peas do we eat? Mostly we eat green peas, right?
Right.
So they must breed for that. Seed coat color, green or white. So here's the color of the seed. Here's the seed coat is green. That's dominant. Okay. Inflated or full pod shape versus constricted, like that, like kind of like a edamame, right? And then pod color green versus yellow, and flower. Oh, not just flower color, purple versus white, but flower position either out to the side or at the very top, axial to the side or at the very top of the plant, which would be termed.
Doesn't that depend on where the sun, like you see if you put a plant somewhere and the sun's mostly on this side that it gets, it
Yeah.
goes that direction?
Not the flower though. The leaves, yes. Yeah, the flower's going to be either side or at the top. Yeah, but I admit I probably wouldn't have noticed. But he was very observant. That's another thing about him. Not only was he patient, he was very observant, and he wrote everything down. So he's a true scientist. people really went back to his work after he was dead and looked at all of his very well written, documented data and were able to repeat his experiments. That's why if you have a lab you've done in this classroom, it should be written, if it were really well written, somebody could repeat it exactly without having to ask you any questions. I don't know that anyone has had that happen yet.
No.
Yeah? No. Okay. All right. Now, y'all have a set of blanks in your book, and you're going to do some matching. I'm not crazy about that, but
Oh, well.
so the first word we're going to be looking at is called the allele. All right, one pair of genes that occupies the same position on a homologous chromosome. Blah. So all those words should be written somewhere, right? And you're going to write a 1 and a blank next to it. Am I correct in telling you that? Is that correct? No.
Kind of confused.
Yeah.
Let's see. Wait, do I have a book that's not the T?
I drew all of these perfectly cursed up.
Are y'all on page 56? Page
That
56.
one, right,
yeah.
That makes sense now.
Does it look like this?
Yeah.
And it says 57? Do you see these blanks going on the left side here?
Yeah.
Okay, do you see this word bank over here to the right?
Yeah.
Guys, everybody, hello,
Yep.
hello. All right, number 1 is allele, and you're going to find, where is it over here? A pair
Third one.
of genes that occupies the same position on the homolog. That's the third blank down. Does everybody see it? Phone
Yes.
away, Asher, now. All the way away, backpack. Did we have a question? Do we see where 1 goes?
Yes.
Okay, thank you,
Ma'am.
thank you.
God.
All right, so what does it mean to have a homologous? Remember when we talked about we have 26 pair or 40, 23 pair or 46 chromosomes, yes?
Yes.
Right, 1 came from your mom, 1 came from your dad. Those are homologous. So let's say we're looking at chromosome number 1. We're looking at chromosome number 1. And on chromosome number 1 of the flowers of the pea plants, the purple flower gene is right there. So on that same allele from the other parent plant, we see it's white right there, okay? So that's all it's saying. An allele is the spot on the chromosome where the trait we're studying is. And the allele is going to be 1 from mom and 1 from allele A, allele B, if you will, okay? All right, and you've got a 1 in the proper deal. So keep in mind, y'all, just because you have your notes written down, like this is the one thing I don't like about the matching deal, is sometimes you write down that number and then you just quit listening to me. All right, but remember, you need to know what it means, not just where it belongs on your page. You need to know what I'm talking about. All right, because in your quiz, you're going to need to be able to answer that. All right, number 2, true breeding. This is what Mendel did to make sure that whatever trait he happened to be studying, like we talked about purple flowers, that he had put enough time into it that he knew the only gene there was for that trait. So if an organism has a certain characteristic that's always passed on to its offspring, we say the organism is bred true with respect to that characteristic, and that is the last one. So you're going to put a 2 there. All right, single trait inheritance. This is just, and we're moving away from our list here for just a second. Okay, so notice here we can see that we have a true breeding here of this green round seed, big R for round, little r for wrinkled. Okay, so is it round or is it wrinkled? If we have true bred for round, and then we breed it with true bred for wrinkled, we're going to end up with 75% round and 25% wrinkled. This right here, this little box to the right there is called a Punnett square, and we will definitely be doing some Punnett squares today on other traits. All right, Mendel's Peas Part 1, we have a little video to watch. Then we'll come back to our chart.
Oh, man.
Mendel believed that each trait of an organism was determined by things he called factors. He further believed that each trait had 2 factors. In the case of the height of a pea plant, for example, there were 2 factors, one indicating the pea plant would grow to be tall. We'll call that factor capital T. The other factor indicates that the pea plant would be short. We'll call that factor small t. Now, Mendel further believed that of these 2 factors, there was one that was dominant. In this case, he believed that the tall factor was dominant over the short factor. This meant in order for a pea plant to be tall, it had to have just one factor for tallness, and that would dominate over any other factor that was there. So for example, a tall pea plant could have both tallness factors. That would indicate the plant would grow to be tall. However, the pea plant could also have one capital T and one small t.
Dominant.
Even though there was a factor for shortness there, the tallest factor was dominant, and the pea plant would grow to be tall because of that dominant factor. Therefore, the only way a plant could be short is if it had two small t's, both indicating the plant would be short. Now, how does this help explain Mendel's experiments? Well, think about his first experiments. In those experiments, Mendel was breeding plants that he said bred true. That meant to him that they had identical factors. The tall plant had both factors for tallness. The short plant had both factors for shortness. Now, when the plants bred, he assumed that each plant gave half of its factors to half of its offspring. So the tall plant would give one of its capital T's to half of its offspring, and the other of the capital T's to the other half of the offspring. The short plant, similarly, would give a small t to half of its offspring, and the other small t to half of its offspring. What do you have in your offspring then? Well, you have each offspring with one capital T and one small t. Since the capital T is dominant, that means each of the offspring will grow to be tall.
All right.
How many times did you say offspring in that one video?
46.
All right. What is a gene? All right. A section of DNA that codes for a protein or portion of a protein, thereby causing a trait. Is that one of our numbers?
No.
Nope.
No? Okay. We learned this before in our previous module because we talked about how it was going to code to make a protein, right? But also that gene is going to code also for a trait. All right. 3. Here it is. Genotype. It's the two-letter set representing the trait. So big T is going to represent tall. Big T is going to represent tall, but with two factors, different factors. Little t is going to represent short. Okay, we'll get to,
Still tall, 'cause 2 negatives make a lot.
no, it's short, sorry. All right, so the two-letter set. So the genotype is the two letters representing a trait. So you have the 3 in the right blank on your page. Yes?
Yes.
Okay. All right, then we get the phenotype. Okay, so the phenotype is what you see, the observable expression of the organism's genes. It's what you see, eye color, hair color, tall plant, short plant. All right, so here we see the genotype is the letters. The phenotype for this would be tall, the phenotype for this would be tall, the phenotype for this would be short. And then there's other factors over here. We're going to see these animals.
Hey, that's what I have. Oh, not blue eyes. I have brown eyes.
Okay. So you understand the difference between genotype and phenotype. Genotype, what are the genes? What are the letters? Phenotype, what does it look like? All right, so here's our genotype. The genetic makeup of the individual. The phenotype is the observable characteristics.
It's a rat.
Number 5, homozygous genotypes. Homozygous, homo means the same. So homozygous genotype is a genotype in which both alleles are identical. Big T, or little t. Those are homozygous. And in the case of big T, homozygous dominant. Homozygous dominant. Little t, homozygous recessive. All right, but they're the same. Here we see big B, and little b. They're both homozygous, one of them for dominance, one of them for recessive. Then we get heterozygous. Hetero means different.
Found it.
All right, so heterozygous is the genotype with different alleles, big B, little b. Little b, big B. In our case, big T, little t. It was still, the phenotype was still tall. The genotype was big T, little t, and it was therefore heterozygous. All right? So we can kind of say, I know it's I've told you several times it's tough to use eye color, but if somebody's got blue eyes, we can pretty much say they're homozygous recessive for blue. Homozygous. If they had any other gene, they wouldn't have blue eyes. You get it?
What if we have, like I have hazel inside of the blue?
I'm gonna look up because I was talking about how David Bowie has 2 different colored eyes. I'm gonna look that up for next time to figure out how that happens. Yes.
So are there any genes that blue is dominant over?
No.
No.
But I think maybe light blue is, so dark blue may be dominant to light blue. Remember, it has to do with that third, there's a third gene. Two of the factors are on one gene, one factor's on a whole other gene, and it influences it. We'll get to that a little bit. All right. Number 7, dominant allele. That's the allele that will determine the phenotype if just one of the one is present in the genotype. In other words, if the dominant factor is present, the dominant factor will be visible. Yes.
Going back to the eye color really quick. My dad also has a weird eye thing. His eyes turn blue when he looks at a person that has blue eyes.
What color would you say?
His eyes, like I've seen pictures of him when he's not like if I'm looking directly at him, his eyes are blue because I have blue eyes.
Okay.
But when he's in pictures and stuff, his eyes are hazel. He
Okay.
has hazel eyes.
I was about to say it's classic for hazel eyes to do that.
But they're blue when he looks at blue eyes.
It's very interesting. It's kind of like also if he wears a blue shirt, they look really blue. If he wears a green shirt, they look really green. That's what I have hazel eyes, and that's what happens to me. Okay. Are we done with our blanks then?
Yeah.
Were there only
No,
7?
there's one more.
Oh, there's one more.
There's 8.
There we go. Obviously, yeah. The recessive allele will be the allele will not determine the phenotype unless the genotype is homozygous. Little letter, little t, short. Little p, white flower. That kind of thing. This is showing the white flower, yeah. Big A, purple flower, big A, purple flower, little a, white flower. All right, we're good with that first set of blanks then, yes?
Yes.
Okay. Mendel's P's, part 2. This is when he's going to take he already had the purebred and the purebred, and he ended up with all tall or all purple flowers. Now he's going to take that second generation, or that F1 generation. Let's see what happens.
Mendel's hypothesis about the existence of factors helped him to explain his second series of experiments as well. Remember, in those experiments, he took the offspring from the first experiments and he bred them against themselves. Each of those plants had one capital T factor and one small t factor. When they bred, the capital T would go to half of the offspring and the small t would go to the other half. So the first plant would give half of its offspring the capital T and half the small t. The second plant would do the same. Half the offspring got the capital T, half the offspring got the small t. Of these 4 offspring in the picture then, their factors would look like this. Now, what does that make the plants look like? Well, the first plant has 2 capital T's. It'll definitely be tall. The second plant has a capital T and a small t. Well, the capital T is dominant, so even though there's a small t there, this plant will be tall. In the same way, the third plant has a small t and a capital T, but the capital T is dominant, so it will be a tall pea plant as well. The last offspring has 2 small t's. No capital T anywhere, so that means it will be a short pea plant. In the end then, 3 out of 4, or 75% of the offspring are tall, and 1 out of 4, 25%, will be short.
So remember, this is the genotype, and this is the phenotype.
They do sound like Squidward.
This is a homogeneous for tall. This is a homogeneous for short. These are heterogeneous. You'll need to know all those things, Joe. All
This
those
Casper.
words. Yes.
There's mirrors that do sound like Squidward.
I'm so sorry.
Does it do sound like Squidward or no?
Okay, I need everybody to
Does that do sound like Squidward or no?
You need to slow down your question.
Squidward. Do you know?
I know who Squidward.
He sounds like it, doesn't he?
Oh,
Like a
the
more
guy
op-
talking does?
Yeah.
Oh, I didn't know that.
Like a more optimistic one.
I thought maybe he sounded more like Patrick, right?
No, Patrick just sounds retarded.
Okay. All right.
It just sounds like
The principles to genetics that we took Mendel's work, and we now have some principles we can write down about them. Number 1, the traits of an organism are determined by its genes, which Mendel had called factors. Do you have to write that whole thing down? Yikes. That's your number 1 point. Is that traits of an organism are determined by its genes. Number 2, each organism has 2 alleles, at least 2 alleles, that make up the genotype for a given trait. There are those weird ones in there too. 3, in sexual reproduction, each parent contributes only one of its alleles to its offspring. And 4, so we have 4 factors here, 4 principles. In each genotype, there is a dominant allele. It exists in an organism. The phenotype is determined by the allele. Now, there are exceptions to these. For instance, there are certain traits that have incomplete dominance. And therefore, there's no dominance of one over the other. So, Oliver mentioned, did you say snapdragons? Snapdragons are one of the famous ones. A red snapdragon and a white snapdragon, and the offspring are pink.
I was talking about like poppies.
Poppies do that too. Snapdragons are the famous ones.
I was saying poppy because like California poppy is orange and red.
And there's incomplete dominance?
Yeah. It's
I don't know about
there's like
poppies.
some is, well, it's like the color, like the color of the singular plant is multiple. Like there's red streaks inside of an orange flower.
Okay. That sounds like they're pretty.
Yeah. It's the state flower of California.
I should make these words a little bigger for you all, yeah?
I'm fine.
I just need bigger pots.
Right.
Just write smaller. Shut up.
Josh, this is coming from you. You have handwriting the size of your ears.
What?
I don't know.
My ears are even big.
All of your ears.
Mine are the same size.
Keep telling yourself that, buddy.
These are small compared
Cut 'em
to
off,
what
it's
I have.
been.
I'm listening now.
You're so big. Fly.
Alright, so, okay.
Oh, okay.
In the World Cup, does FT mean final?
Yes.
Yeah.
And the USA won.
Yeah.
Oh, sweet.
Let's go.
England beat
Done.
Wales. So what happens now? England beat Wales 3-0. That does matter.
Well,
Cool.
two teams from each group will move on.
Will that be USA and
USA and
UK?
England will move on.
That means round 2.
Alright, are we good? Everybody got this?
I have one more.
Okay.
I want to know who they're going against in the brackets.
We won't know that until the rest of the games are done, matches are done, whatever. They call matches.
I still think it's funny that Qatar lost their host.
That's not surprising. They're terrible.
They are very bad.
How'd they make it?
How'd they get the games there? I heard it was bribery.
It was
Oh.
very,
That's what I heard.
yeah.
I don't know, I'm not a big.
Argentina is gonna beat us all, just saying.
Oh yeah?
Yeah.
They just lost to
I was about to say
I know.
they lost to somebody.
They lost.
That means they're out, right?
to
That means they're out?
Saudi
No.
Saudi Arabia.
Yeah, which was a very disappointing game for
I'm going
so
for
many.
Spain.
Messi, come on, Messi.
Never watched soccer before, so I
Messi's
don't know.
not Argentina.
Yeah, that's
He
what I'm
is.
saying.
He's Spain.
Spain.
I thought he was Argentina.
That's Ronaldo.
Man.
I'm kidding.
No,
I'm kidding.
but that's
I'm kidding. I know.
The
I'm
actual
kidding. I
football.
know. I wanted to say that just to sound kidding.
Okay, I have to move on, y'all.
Wait, I
No.
said formal words.
I'm good.
Ronaldo and
How do
Messi
y'all
might
write
be
so
better.
fast?
No, I thought they were friends.
They're kind
My
of
finger
frenemies
hurts
at
from
this
writing.
Because they're both it's Portugal and Spain, they're the same thing. Just one is a slightly different language.
What did you say?
You said Portugal and Spain are the
I'm done.
same thing.
Who's Casper?
No, you're not.
He just said Portugal and Spanish are the same thing.
That's
They're-
true.
I said Portugal and Spain are practically the same thing. It's just one is a different language. They have very, very.
That's not the
similar-
same thing then.
They have
They're
very,
both
very
on the
similar cultures.
Iberian Peninsula.
They both have very similar cultures. They did the same thing.
Isn't
They
it
do
about
have
like Canadian
different
American.
languages.
American?
Okay, there we go.
thing?
Alright, y'all, so this is called a Punnett square. Y'all are gonna have to become proficient at doing a Punnett square. For any single definable trait, we can predict the likelihood of the offspring given the genotype and phenotype using a Punnett square. So whatever this is a pink flamingo and a blue flamingo, I guess. I don't know.
It
Doesn't
looks
matter.
like an emu.
So we have a homozygous dominant trait for red or pink. We have a homozygous recessive trait for blue. Now what I'm going to do is I'm going to pull this A at the top of this box down to both boxes. A goes down 1, A goes down 2. Big A, big A. Same here, big A, big A. I'm going to pull this A over twice. I'm just distributing those in this box. Does that make sense?
Yes.
So now what do I have? What are my offspring? They are big A, little a is their genotype. What is their phenotype? What do they look like?
They're red.
How many?
All of them.
100%, right? 100% red, because there is a dominant big A in every one of them.
Domination.
Yes.
It says that there's a reference on page 264. There's no
Of
page.
the textbook.
Oh, okay.
All right. Next, we're gonna see. Oh, what am I doing here? Let's see. Do I have a? I do have a video.
In this example, we're breeding a homozygous purple flowered plant with a white flowered plant, and we're asking what are the possible genotypes and phenotypes of the offspring. Well, to figure this out, we first have to determine the genotypes of the parents. Let's start with the homozygous purple flowered plant. Since it produces purple flowers, it has to have at least one of the dominant alleles, capital P. However, it's homozygous, which means both its alleles are the same. Therefore, the only possible genotype is capital P, capital P. We put those two alleles at the top of the Punnett square to represent that parent. Now let's look at the other parent. The other parent produces white flowers. That's a recessive trait. To express the recessive trait, you have to have both recessive alleles. So the only possible genotype for the white flowered plant is little p, little p. We'll put those two alleles to the left of the Punnett square to represent that parent. Now to figure out the offspring, all we have to do is fill in the boxes. Let's start with the columns. There's a capital P on top of the left-hand column, which means a capital P goes in each box on the left. There's a capital P on the right-hand column, which means a capital P goes in each box on the right. Now all we have to do is look at the rows. There's a small p to the left of the top row. That means each box in the top row gets a small p. There's also a small p to the left of the bottom row. That means each box on the bottom row gets a small p. In the end then, we filled in the boxes by simply looking at the letter at the top of the column and to the left of the rows. Each box now represents a possible genotype for the offspring. But look at the boxes. They all have exactly the same genotype, capital P, little p. So, all offspring will have the genotype capital P, little p. And since this genotype has a dominant allele, capital P, that means all offspring will produce purple flowers.
But all offspring also are heterozygous, right? Big P, little P. They have different alleles.
Miss Casper,
Yes.
that seems really easy to do, actually.
Okay. Did it?
Yeah.
All right.
Because you just gotta put the big one.
Absolutely.
You
So I
gotta
don't
put
want
top
to
in
have
front of it.
anybody scoffing when I give you a quiz question that asks about Punnett square if it's easy. Okay? Here we go. Example 7.1. Oh, we just did that. We just did that. Yeah. I don't know if we're using F
So
here.
that'd be big F and little f in all of them.
100% purple flower. We should have done something besides purple flower.
What was that?
Crazy. All right. But
Oh, I
what
got it.
if I have, listen.
Wait, do we write that down?
You have to write that one down? Go ahead.
Oh wait, I'll do it.
I don't know why they're using F. I have no idea.
No, there's no point in using, I mean, it just says, give an example of it. So, I guess we could use any
Which
letter.
does point out it is arbitrary. I can pick, all we know is that the dominant needs to be uppercase and the lowercase is the recessive, okay? It doesn't matter what the letter is that we're using. We could use big Z, little z. I would use letters that are easy to tell the difference between big and little. You know, if it's
J.
S and S.
B and
What about
C is
L?
big and
What about L?
B is a
B
good
and
one.
L. Big
L
and
is pretty good, pretty big difference between uppercase and lowercase. A is pretty good difference.
Does it matter
Just put,
what
like-
order they are? Like if it's
If it went little f in front of big F, no, that doesn't matter. It's just traditional that we put the uppercase letter first. Good question. Adrian, question?
So do we just like put 100% underneath?
So then we're gonna have 100% purple flower and 100% heterozygous. Right? So now we're gonna take this generation, F1 generation, we're going to mix those, okay? We're gonna breed those. Are we ready? So I have big F, little f going across. I have big F, little f going down. So now I'm gonna distribute those. Here's my big F, big F, big F, little f. Now that bothers me. The F should be blue and the little f should be red, right?
Yeah.
Big F, little f, and little f, little f. What's happening now? We have heterozygous purple flowered plants, heterozygous purple flowered plants. And our offspring are 75% purple, 25% white. But we can further define that as 25% homozygous purple, 50% heterozygous purple, and 25% homozygous white. All right, got it? And you've got your boxes filled in there? Okay. All right. A test cross is a breeding of an organism displaying the dominant phenotype but unknown genotype. Unknown genotype. We don't know if it's big T, little t, or big T, big T. We just know the dominant trait is showing. All right, but when we go through a breeding of this test cross, we will find out pretty quickly whether or not we have 100% of dominant gene or only 75%. Okay? As long as we know what we're breeding it to, usually a true bred, then we will be able to find out. So more mono-hybrid crosses. This is just using one trait. I have big A, big A, big A, little a. I'm going to bring these in like this for a ratio. Notice I have 50% homozygous dominance and 50% heterozygous dominance. Whatever A is standing for, A is showing. All right, it is expressed. % dominant is our ratio. Do y'all have boxes for these two? On the next page?
Yep.
Everybody got it? Chop chop, it doesn't take long to write those letters down. Next we're going to try big A, little a, and little a, little a. That gives you a good idea. We're probably going to have some recessive expression now. We're going to bring our big A down, our little a over. Notice what I have here, 50% dominant, 50% recessive. This, by the way, is what happens when we're talking about a baby being male or female. It's always 50%. 50% XX, 50% XY. Every time.
Anyways, I thought like it was like slightly more for a female.
Nope, 50-50.
Oh.
We have slightly more females being born right now, but it's still 50-50 chance.
Yeah,
Ratio's same.
I read somewhere, it was either inside my textbook or like when I was studying it, but it was like there is like more like males because they are easier to like die or something.
Dang.
Like,
That is very true.
Born
But
or
like,
something.
When it comes to the distribution,
No, but I was saying like yeah, I read that. Maybe that was wrong.
when it comes to the distribution, it's 50-50. Afterwards, there is right now a
most guys
no.
are
I'm just
stupid,
saying
so we die.
environmentally there might be something to change.
That's
Right
very
now,
offensive to me.
we happen to be the tendency happens to be like a little over 50% females being born, but it doesn't really mean anything genetically.
I like those sports. No,
Okay.
you can all be
All right, so
smart.
here we're going to see, this is dogs expressing blue eyes. You know, dogs will have blue eyes. And then there are those dogs that have one blue eye and one brown eye, right? So we're going to see that the dog named Carrie has brown eyes.
Carrie.
The dog named Rusty has blue eyes. They're going to breed them together and get 50-50. 50-50 chance of blue or brown eyes for these dogs. All right, Punnett square 1, in certain breeds of dogs, deafness is due to a recessive allele, little d, of a particular gene. And normal hearing is due to its dominant allele, big D. What percentage of the offspring, listen, what percentage of the offspring of a normal heterozygous dog and a deaf dog would be expected to have normal hearing? So what we're going to do is we're going to take the big D and put it up here, and the little d here, right? That comes from right there. And put the little d and the little d. Now, surprisingly, gang, listen, this is where you make your first mistake. I have students trying to do this, trying to put the d's like that. Okay, that's wrong. That's wrong. You're just going to put one of the alleles over each box, okay? So, a little d there and a big D there. Got it? Okay, so what's going to be this first box down?
Big D, little d.
Big D, little d. The next one down. This one coming over here. And this side? How many dogs can hear?
50%.
50%. How many are deaf then?
50%.
Very good. But because I'm making it look so easy, you do need to practice, okay? Because you're going to end up making that mistake, okay? Don't do that. All right, in man, normal pigmentation is due to the dominant allele we're going to call C. Although C is kind of tough because a big C and little c are very similar looking, right?
It's Asher.
All right, listen.
I wish.
There is a condition called albinism or albinoism that is due to a recessive allele little c. A normal man marries an albino woman and their first child is albino. That tells you something about the man. He had to have an albino gene, right? He had to. Otherwise the child could not exhibit that. What are the genotypes of these three people? If there are more children, what would their probability be like? Okay, well, we already know mom for sure is an albino, so we're going to say little c for mom, right?
Does albino?
Would it be an A because it's albino?
That means they don't have any melanin, any pigmentation in their hair, eyes, skin. They're completely white.
Oh,
Wouldn't it be A if it's albino? Isn't it?
It told us to use C.
Okay.
Yeah. I don't know why.
Interesting
So
enough.
what do we know
Because
about
the
the
child.
dad? The dad does not have albino or albinism, so we know he has a big C, but he's had a child with it, so he must have the gene. He must be heterozygous. So our first box is going to be big C, little c, big C, little c, little c, little c, little c. So what about their children now? Their children they have 50% chance of their children having albino.
Miss Casper,
When
when you
you
step in
walked
front
in
of the
front,
projector,
yeah.
the dude's face was on your back.
But he was all goofy looking.
Crinkly.
We should totally, for Christmas, we should all make her a lab coat that has that on the back.
Okay, Punnett square number 3. In minks, brown is dominant over silver blue. What offspring would you predict if you crossed a homozygous brown? That's going to be big B, big B. Homozygous. He's brown. With a silver blue, it's going to be little b, little b. What's it going to be, gang?
It's gonna be 100% brown.
100% brown. Big B, little b in 100% of the boxes. Yes.
So what if it's
Monkeys.
one has these genetics of like brown and silver, but another has the genetics of like
This is a mongoose.
silver and white. So what happens there?
When we have a third thing coming in like this, I have 3 pictures right
Yeah.
here. Okay, that gets more complicated. All right, so we have to figure out where is the gene being distributed. Okay, is it on a whole different gene or what? Yes.
What happens if there was like an animal that, like, species that was going extinct and they only had a certain amount left, and they all weren't, none of them were like purebred or anything?
Okay.
Would it ever get back to purebred or like never?
I don't know because like they tried to get the DNA from like a woolly mammoth and implant it in an elephant, right? And it would be similar, right? So I don't
Would it
know.
ever get
They
back?
can try. They can try. And now that we have the ability to do cloning type things, we can maybe do a cloning. All right, now here's something. This is what I was mentioning earlier. In snapdragons, R is dominant over pink. So what would be the phenotypes with that? Okay, we know we're going to have. What does it tell us? Heterozygous red will be big R, little r. And we have little r, little r. So we have big R, little r, big. Are we back to 50-50 again?
Yeah.
So 50-50. So the Punnett square is just like this. Y'all feel comfortable with these Punnett squares? Good deal. I will tell you this, that if you do breed a red and a white, you're going to end up with a pink because there's incomplete dominance. Okay, that's a Punnett square. This right here is called a pedigree. A pedigree is where we can look at something from a distance and right off tell what's going on. Okay, so a pedigree is a diagram that follows a particular phenotype through several generations. If you're trying to track something, say in a family, a disease or other trait in a family, we can look at it at a deal here. What we're going to see here, one thing you need to know, a square represents a male, a circle represents a female, colored in represents the trait being expressed, and blank, no color in it, is they do not have the trait. So for instance, we're going to see here, whatever this trait is, one of the sons ended up with that trait, two of the children, this son and this daughter, did not end up with the trait. Okay, so we can probably make a few, if we can probably say that whatever the trait is probably recessive, right? That's not a whole lot of data. We need more data than just 3 kids. We need like 100 kids to get really good data.
Oh my God.
Easy math, okay? Right here though, you can also tell if that is expressed in that generation, then dad must have had the gene there. Must have had the gene. Okay? Alright, so let's look at it here. We have, I don't know what the trait is once again. We're going to see mom and dad. Here's mom's got it, dad doesn't. And some of the children got it. A male and a female. Okay? Notice down here.
I think they had 12 kids.
No, it's generations.
Yeah. So, this is a daughter of these, this is a son of these, this is a daughter. So these ones coming off not connected to this bar, this one's married to this one's married to this one. Alright, so in this case, this daughter married this guy and none of their children had it, so they probably don't have the recessive
They've been
gene.
discontinued for the
In this one we have, all the children got the gene, whatever it is. And in this one, half of them got the gene, whatever it is. Okay? Alright.
They had 3 kids and each one had 2 kids.
Video.
Kind of like that.
In this example, we're looking at a pedigree for a type of animal that can only have blue or brown eyes. In pedigrees, squares represent males and circles represent females. That's not important in the analysis, it's just a standard. In this pedigree, if the square or circle is filled, then the animal has brown eyes. If it's hollow, the animal has blue eyes. The goal is to figure out the genotypes of the parents. That's actually not very hard. Let's look at parents 3 and 4 to begin with. Both of these parents have brown eyes, but look at the offspring. 2 of the offspring have blue eyes. How is that possible? In order to get a trait, you have to receive an allele from a parent. So these parents have to have a blue eye allele. They don't express it, however. What does it mean when you have an allele that you don't express? It means the allele is recessive. So the blue eye allele is recessive, and we can use a small b to represent it. If the blue allele is recessive, that means the brown allele is dominant, and we'll use a capital B to represent it. Now we can figure out genotypes pretty easily. We've already determined that parents 3 and 4 each must have a blue eye allele. After all, some of their offspring express that trait, and the only way you can express a recessive trait is to get one allele from each parent. So each of those parents has to have a recessive allele, but neither express it. That must mean they each have a dominant allele as well. So each of those parents must have the genotype capital B, little b. We can also figure out the genotype of parent 2. After all, she expresses the recessive allele. The only way to do that is if you have 2 of the recessive alleles, so her genotype must be little b, little b. Well, now all we have to do is figure out the genotype for parent number 1. Well, look at the offspring. None of the offspring have blue eyes. So most likely, parent 1 doesn't have a blue eye allele, or at least some of his offspring ought to get it. Since none of his offspring got it, he most likely doesn't have a blue eye allele, so his genotype must be capital B, capital B. Think for a moment about how much you can learn by looking at a pedigree. By looking at 2 families, we've already been able to determine the genotypes of the parents. That's a lot of information.
All right. More complex crosses. We're going to see where we're going to follow more than one trait. We can follow 2 traits. Looking in this particular case, it's round and yellow. Round and yellow. Round yellow, round green, wrinkled yellow, wrinkled green, so we can do that. No. So, but if I'm doing that, you're going to see what I have, what is going to go in this box right here for 2, I'm following 2 things, big F and big T. So let's say purple flower and tall, white flower and short. When I put it in there, you see I'm going to have those 4 things right there like that. Okay? That way, the genotypes are 100% big P, little p, big F, little f, big T, little t. I don't know why I have an F in there. For 100% tall, 100% purple, because the parents were homozygous, and this 1-to-1 Punnett square. Punnett square is easy, but we're going to do it. Y'all have a homework thing to do, and it's using big F, big T, little t, showing height and color, it's gonna be something that with 16 boxes in it like this. And it's gonna look like that, and that's all I'm gonna show you.
Oh my
The
god.
rest you have to do on your own, and you have to come up with the ratios, so the percentages of whatever of the genotypes and the phenotypes. This is a homework, and I expect it to be done by next week.
It's right here in the book.
Better write the letters.
It's really not a difficult thing.
It's
but
not
It's
difficult.
not difficult. The ratio part is difficult.
I think.
What did I do that for?
I think this is the easiest part of this entire class, in my opinion.
Yeah. Except for filling out the study guide.
Okay, so you end up with 16 boxes. All right, and now what we're going to do, it's time for a lab. We're going to do a genetics probability lab. We're going to be working
is the thing
in
in our
groups.
books?
It is.
The homework thing?